Now look at this.
What's up about penguins? So today we're going to do topic 2.11, origins of cell compartmentalization. So prokaryotes we know are significantly smaller and eukaryotes we know are significantly larger. And we talked about in the last video that it's due to the compartmentalization. So there's more compartmentalization in a eukaryotic cell than there is in a prokaryotic cell. This lack of compartmentalization in a prokaryote is one of the reasons why we see transcription and translation occurring simultaneously. In a prokaryote, they have a nucleoid. This is a region where the DNA is going to be found. So as soon as it transcribes and makes that mRNA, the ribosomes from the same area can immediately translate that mRNA into a protein. Whereas in a eukaryote, we're going to see transcription occurring within the nucleus. It makes the mRNA. Then the mRNA gets modified, and then it will leave the nucleus to find the ribosome to be translated. So though we've mentioned these differences, there's a lot of similarities. So both a prokaryote and a eukaryote are going to have ribosomes, and those ribosomes allow the cell to go through these different processes of protein synthesis. They also are going to have a plasma membrane. That plasma membrane allows them to have a different internal environment than external environment, so they can have all these reactions occurring inside of their cells that aren't occur outside their cells, or they're able to have a difference in concentrations, and there's all these different things that they can have due to that plasma membrane. So then how did we go from a prokaryote to a eukaryote? Endosymbiotic theory. So there's this theory that states that back in the day, there was this ancestral prokaryote that engulfed a chemosynthetic prokaryote, right? And so when it engulfed that prokaryote, it then made this symbiotic relationship with it. So the chemosynthetic prokaryote provided ATP to the ancestral eukaryote, and the ancestral eukaryote provided the sugars needed by the chemosynthetic bacteria. And then after that lineage, there was a divergence in which there was one that engulfed a photosynthetic prokaryote. And that's why we would see that all eukaryotic cells have a mitochondria, but not all eukaryotic cells have the chloroplast, which means that the mitochondria was engulfed first. And this diagram kind of shows a little bit better. It shows the invagination that occurred at the beginning, which created a lot of those internal membranes. And then you also see, of course, the mitochondria being engulfed first, and then you see that chloroplast. So what evidence do we have that proves this? So if we think about the way that mitochondria and chloroplasts reproduce, they reproduce independently of the rest of the cell. They go through binary fission, just like a prokaryote. The ribosomes in a mitochondria and a chloroplast are the similar size that we see in a prokaryote. They also have similar DNA. So the DNA in a prokaryote we know is circular, and the DNA inside of a mitochondria and a chloroplast is also circular. Bye, y'all.